typical episode uh, where our typical episode consists of stiffening and flexion position and the face turning pale, inconsolable screaming, and finally loss of consciousness. Now you see this redness is hemicorporeal erythema. It splits from one side to another side, and it can be triggered by putting, for example, a temperature probe in the anus or defecation. So again, a very dramatic pain syndrome arising from a single snip. So that brings us to the question that if SCN9A and sodium 1.7 channel is so important to gain or loss of function of pain, surely there can be a cure found on this basis. But unfortunately, most of the cures we have looked for are blockers of NAV 1.7 or antibodies to 1.7 have not worked out. So that means that the problem here has been the selectivity of these blockers to exclusively sodium 1.7. And I think there are studies going on to explore the role of tarantula toxin, for example, which has got a high selectivity for NAV 1.7, and gene therapies for NAV 1.7 blockage. The other targets around NAV 1.7 are NAV 1.8 and NAV 1.9. Now, that is from basic research, and you can't possibly do much about those conditions. But what can we do today using genetics at the bedside? Now, I will point you to this article by Craig et al. In British Journal of Pain in 2013, which is probably one of the very good articles on pharmacogenomics. And essentially, when we take our painkillers, they have a pharmacodynamic effect and a pharmacokinetic effect. And this diagram shows you some of the genes which have been found to be hugely influencing this process. So, for example, the most important gene in this category is CYP2D6, which codes for the metabolism of codeine, tramadol, and oxycodone and hydrocodone. Phase 2 metabolism of morphine is coded by UGT2B7, whereas the opioid receptor is coded by OPRM1 gene. And the ABCB1 is a very interesting mutation which causes increased efficacy of morphine in cancer patients with decreased central side effects. So there are lots of opportunities here to refine how we use our analgesics. And this study here shows, I'll just go talk to you through this slide. This is a study which used CYP2D6 guided analgesia use. And so IM stands for intermediate metabolizers, P stands for poor metabolizers, and NM stands for normal metabolizers. So the black box shows the change in pain intensity at 3 months in the group in which analgesics were used using the algorithm defined from the genetic profiling of CYP2D6, and the pain intensity was better in that group as opposed to in whom genetic profiling wasn't used. So based on that study and some others, the authors have come out with now a guidance as to how we should do the genotyping of these CYP2D6 and classify them into phenotypes of ultra-rapid metabolizers, normal metabolizers, intermediate metabolizers, and the clinical recommendations which flow out of them. Then, apart from, again, single mutation studies, another good way of studying genetics in the past has been linkage studies. That means if two genes are quite close to each other on the same chromosome, and they are studied together, for example, G6PD gene, and along with one of the other clinical conditions, is studied together, and the other gene is looked for in the linkage study, and G6PD deficiency is inferred from that. From the point of view of pain, I could pick up this example of ankylosing spondylitis and inflammatory back pain, genes being linked together. Another thing which you need to understand when you're trying to understand the genomics is the role of pleiotropy. That means one gene has got many roles. So, for example, pleiotropy is seen in this gene on chromosome 12, SLC16A7. And this gene is associated with the development of both chronic pain and major depressive disorder. Now, I've highlighted the frizzle gene in the chicken, which means that this gene codes for this curvy feathers rather than the flat feathers, but it also increases the metabolic rate of this chicken, and this chicken actually lays less egg on a daily basis than the normal chickens. So, while we tend to get carried away by single gene inheritance and its role in chronic pain, I think more and more in the future, we're going to see the role of polygenic inheritance. That means many genes coding for a single clinical trait, such as chronic pain. So for example, I've showed you that there are about 11 genes coding for hereditary, sensory, and autonomic neuropathy. And the ones which we might end up seeing sometimes in a pain clinic would be the HSAN3 and HSAN4, which have pain issues along with autonomic instability, sometimes mental retardation, and other issues like hyperhidrosis. So that brings us to the study of genome-wide association studies. So far I've talked about single mutations, I've talked about linkage, I've talked about polygenic inheritance. So genome-wide association studies are based on the fact that you have cases, for example, chronic pain, and then you compare them with controls in the population. And then along the x-axis are laid down all the chromosomal genes, and along the y-axis is the log value, and there is a dotted line, which is the p-value of significance. So all the dots which you see above the dotted line are significantly associated with chronic pain here. For example, 5 of these genes are associated. Now, most of these are still single nucleotide polymorphism, right? But it gives us an idea which are the genes which are associated with chronic pain and what do they code for apart from pain, let's say a nerve channel or anything related to the physiology of pain. Are there other expressions of this gene? So it is an exploding area in chronic pain. And at last count, we have got about 194 associations for migraine, about 36 for temporomandibular disorders, 29 for fibromyalgia, 20 for low back pain, visceral pain, cancer pain, and so on and so forth. And Diachenko, again, as I mentioned earlier, is leading the charge in this area of thrust. In the UK, there was a study carried out in the UK Biobank. Now, UK Biobank is a conglomerate of about 500,000 individuals who have agreed for their genes to be tested and have been recruited between the ages of 40 and 79 between 2006 and 2010. Now, in this particular study, the phenotypic trait of chronic pain which was tested was multi-site chronic pain. That is pain in more than one site to up to 7 sites. This is, and they excluded whole body pain like fibromyalgia, etc. And they found 113 genes across 39 genomic loci which were associated. Now, I might warn you here, association does not mean causation. It means association. So one has to be very careful here. Apart from GWAS and the hardware, let's say, apart from the fact that we inherit hardware and which increases sometimes our susceptibility to chronic pain, are there environmental influences which also affect the genetic machinery to make us more vulnerable to the pain intensity and the feelings of pain. So to understand that, let's go back to the basics as to how epigenetics is studied at the genome level. So if you have an environmental influence on the genes or the DNA, it shows up either as DNA modification or chromatin or histone modifications. Histones are the scaffolding around which our DNA is wound in the nucleus. It will also show up as variations in non-coding RNAs. Remember that part which we said was junk DNA, was coding for RNAs which did not code for proteins. And there seems to be a variety of these non-coding RNAs, some of which are affected by epigenetics and their modifications. For example, micro-interfering RNA, which I've shown here. So chronic pain, so far, emerging field, not much of work has just been done. One of the genetic correlation or association has been found between histone decitylation and central sensitization in chronic pain. So that brings us very quickly to this woman I was talking about right at the beginning of why I dived into genetics despite being a jobbing anesthetist. And this paper was published on 29th of March 2019, and that was also the day I had to attend a Jeremy Vine show, and I was being quizzed after Theresa May had been interviewed, and this article had come out in the Times that day, which sort of did a wordplay on Our Lady with No Plane with Theresa May, and with the knives in her back causing her pain. Okay, from there we come to, if we want to understand what happened to this special patient of mine, we have to understand the genetic architecture of a gene. So there is a coding region which consists of exons and introns. The exons are green, the introns are blue. Now, there is a promoter region which promotes the expression of this gene and can also stop the expression of the gene, and there are upstream enhancers. These form the RNA, and the final RNA produces the protein. So in this particular patient, which I'm talking about, in chromosome 1, we had a single nucleotide polymorphism. PFS is the index patient here, and the son is a partial phenotype. So both of them have this deletion in the non-coding portion of the DNA. And this particular stretch almost is similar to this stretch which codes for Fa. Fa is fatty acid amide hydrolase enzyme, and that metabolizes endocannabinoids like anandamide, PEA, OEA, etc. So this has got a S and P here, single nucleotide polymorphism, beg your pardon. And this has been seen before in breast surgery patients, that if you have this SNP, you have less morphine requirement. But this deletion, along with these regulatory sequence, these are called ALU sequences, which predispose to genetic changes, were not seen before. Okay, so this gave rise to, then we further did the analysis of the endocannabinoids in red in the patient against the controls, and we saw that AEA, PEA, OEA, and 2AG was raised. So then we need to understand what is gene therapy, which means that we use genetic seizures to recreate that particular genetic defect, which is very useful from the pain point of view. And this is an emerging therapy in pain management. And we have already, my collaborators in UCL have used the genetic scissor to mimic the far-out microdeletion and in the human cell line, and they have found a reduced expression of FA. And we're now progressing to animal experiments pretty soon. But this is just the beginning of the genetic journey in pain management. We are out here. We know the phenotype. There's a lot of phenotype epidemiological work being done. We are beginning to unravel the genome, but we just don't know much about transcriptomes, proteomes, metabolomes, and brain imaging is again joining up with the phenotype here. So all these, when these ducks are lined up in the future, you may get viable drug targets. So in the future, I think more work will be done around the fact that we will be trying to link polygenic pain inheritance with the epidemiological chronic pain phenotypes. And we have been helped by the ICD-11 in the definition of these phenotypes. Now, in my case, for example, the gene which coded for Phi enzyme and gave extraordinary pain relief. So that set us thinking about the role of endocannabinoid and endorphin systems and the interaction, and research work is now following in that direction. There'll be growth of pharmacogenomics and genetics to tailor existing treatment. We shall probably be getting genetic data at the bedside regarding our painkillers in the near future. There has to be more investigation of non-coding DNA and RNA. And again, there has been an imbalance in genetic research, in that most of the genetic research at the moment is being carried out in white Caucasian populations, and we need to have more equity on that. And we need, genetics is there, and it is there to stay, and it will form a very big part of a clinical practice in the future. So we need a structured teaching program for clinicians. With that, I would like to end, and thank you very much. If there are any questions, I'd like to take them now.
Dev, I think that was brilliant. Thanks very much for that. I mean, I think it's enlightened me about the importance of the non-coding part of the genome, which I really didn't have much of an idea about at all, to be honest. And I quite enjoyed the fact that lots of SNPs aren't important, and that kind of grey coat goat thing was quite a good thing, wasn't it? I think the delegates found that also quite interesting as well, and they've got a few questions. One was, are there any kind of specialists centres in the UK that with look at genetics and pain, or is there a kind of lead centre in the UK for genetics and pain?
Yeah, if I were to answer that, I mean, when I had this patient, I went around shopping. So it landed me with Marshall Dever in Israel first, and then with Stephen Waxman in US. And then finally, I was pointed to UCL, where John Woods and James Cox and Geoff Woods from Cambridge as well. And these are the three, I mean, that department in University College London is probably the leading pain molecular biology center probably in the world, probably.
Right, okay. UCL is the place for the UK then, it seems. Okay. So what do you think? Obviously most of us are kind of jobbing pain consultants, and we're interested really in what the first steps practically are going to be in genetics and chronic pain. Where are we going to see genetics come in a day-to-day, in our day-to-day practice? Where are we first going to see its impact, do you think?
I think we already, in the next few years, we're going to see it first in the analgesic management of opioids. And that would be probably, and genetic testing to predict who would benefit from opioids, and how the efficacy of opioids from the metabolizers, normal ultra metabolizers, etc. I think these are the low-hanging fruits from genetics straight away. Yeah. But in the longer term, I think we've got our act together as far as the phenotype classification of our genetic of chronic pain conditions are concerned with the ICD-11. Now we can start associating genes with those conditions. The idea would be to then start working backwards on the pathophysiology. For example, from my case, you have to work on the pathophysiology interaction between endocannabinoids and opioids. But that is research. But the clear thrust straight away would come from one area would be the pharmacogenomics. And the other one, if the gene therapy things succeed in the near future, and we might see a few gene therapies of, for example, of the dorsal root ganglion.
Okay. The tests for trying to find the mutations in the genes to encoding the CYPD6, for example. Are they easy tests to do? Are they quite quick tests to do?
Absolutely, absolutely. These have become so. I mean, in 2003, it used to take over $1 million to sequence a genome. Now it takes less than $1,000, and lots of companies have mushroomed, especially in China, which would do it for maybe a few hundred quids.
Right. Okay. One thing was the twin studies you saw that you could look at, how heritable conditions were, looking at monozygotic and dizygotic twins, and it was clear that things like IBS weren't as heritable as things like rheumatoid arthritis. And I think it's a 59% heritability. I mean, do you know? I was a bit unclear about what that number kind of meant, really.
Now, if you dive deeper into that article, you see some of these studies are quite dated.
Right.
So now, if you remember when we presented the whole genome in 2003 as having been sequenced, that was not really true. 8% has still not been sequenced then. And that work was finished only last week, right?
Right.
Our genome libraries are still being built up, right? So I think there is going to be a period of consolidation of our genome libraries before we can, conclusively. I think we'll have to revisit those estimates again.
Okay. I was just trying to get an idea of what those numbers meant, whether the twins.
But they give you an idea based on current knowledge.
Okay. I think it's given us an idea that which conditions are more heritable anyway. Fine. Just looking through the chat box a little bit now, if that's all right. So, any examples of targeted gene therapy in chronic pain management in current practice? So, gene therapy. Is any gene therapy going on for chronic pain management at the moment?
I mean, at this point of time, we've had animal experiments which have shown that if you target the dorsal root ganglion and then you can manipulate the pain outcomes in animal studies, and this is using vectors called as AAV or adenovirus vectors with CRISPR-Cas9, which is the genetic scissors, and targeting some of these genes like NAV1.7, etc. The team flowing out of our study, the team at UCL is working with global consortium now to develop a gene therapy based on our findings. But as of now, there are no clinical gene therapies available, but they're available for other conditions, medical conditions.
Yeah. It's quite an exciting time, really, isn't it? It's really exciting. So, one other question. Are there any UK plans for pharmacogenetic passports like in the Netherlands or Denmark? I'm not sure if,
No, I do understand that question, and that's a very important question. So it is essentially like you have malignant hyperthermia and you have a genetic testing, for example, and you carry that passport. Similarly, do you understand? Do you get that? Yeah. So I think a similar thing. I think we are at a very early stage to have definitive passports for pain management, but I think it's a very viable idea for the near future.
Yeah, that would be useful to know, actually, wouldn't it? If there's any point giving someone codeine, for example.
Yeah.
Okay, lovely. Fine. I think I've asked most questions on there. I think there's lots of praise for that talk. I think that's pretty much it, Dev. Thanks very much for that. I think I found that really interesting.
Thank you, John, for indulging me, and thank you to the audience for giving me a time. See you. Bye bye.